today's theme is explore, and we're specifically going to focus right now on explore your genome. So what does it mean to look into your genome? So we're going to spend a few minutes walking through this. And hopefully I'm going to meet Glenn's #1 rule, the first one he put up, to make learning fun so that nobody is twitching and in fear in memory of a difficult organic chemistry class or genetics or biology. So let's start. We're going to talk about understanding your genome for a number of different things. We're going to talk about how we understand history from it, health, a little bit of humor along the way as well. So let's cover some basics. So as Amy said, every organism is composed of cells. In humans, we have about 100 trillion cells. And every one of those cells is controlled by a set of DNA inside that cell. You can think of it as a set of instructions, a master regulator. And so the genome is the collection of all the DNA that's in an organism. You have a yeast genome, you have a dog genome. We're going to talk about the human genome. And there are individual pieces of this genome that tell the cell how to make things. And so those are going to be our genes. So genome and genes. Here's another way to think about this, especially for those of you that like to cook. So the cookbook is our genome. That contains all the recipes, which are our genes. And those recipes tell the cell how to make things. Now, our cells don't normally crank out chili and pork tenderloin and chocolate cherry cupcakes. They crank out things like collagen and hemoglobin and insulin. But the analogy still works. What I want you to remember is that in this cookbook, not only do we have the recipes, but we have a whole lot of contextual information that says, here's what you should ask for when you go to the butcher and ask for a certain trimmed tenderloin. Or here's a wine pairing that works well with this menu. Or here's how you set the table appropriately. So all that contextual information is in the genome, not just the gene recipes. In fact, the recipes only make up about 1% of our total DNA. Okay, DNA has 4 chemical building blocks, really easy to understand. They're abbreviated, we call them adenine, thymine, cytosine, and guanine, but for you, you can think of them as A's, T's, C's, and G's. So when we talk about a genome, this is what we're talking about. A whole string of letters. Now, for you and I, this just looks like a bunch of headache-inducing letters. But for the cell, our cell can read through this and go, oh, well, right here's the instructions for how to make collagen. And oh, look, down there, there's some insulin and hemoglobin. Now, there are 3 billion letters in the human genome. So if we were to put these into phone book kind of format, the phone book stacked end to end would be as high as the Washington Monument. One cell's genome. 3 billion letters, Washington Monument phone book. We have a lot of information that we have to work our way through as our cells. Everybody with me so far? Yes, thank you. I need you to verbally speak because I can't see any of you. So the words are good. That's good. All right. If I were to take any 2 people on the planet and I were to look at their DNA and start sequencing through all 3 billion letters, 99% of our DNA is exactly identical. Let me say that again. Any 2 people on the planet, 99% identical. Pretty amazing. But we do have differences. So what happens with those differences? Well, sometimes, let's back up. Sometimes those differences make no change at all. Like I'm going to buy white lily flower instead of swan's down. Probably not going to make a big difference. Sometimes those changes lead to normal variation. Differences in our eye color, our hair color, our skin tone, our height, our body shape. That's just part of the normal spectrum of what makes humanity such a beautiful rainbow. Sometimes this variation leads to increased disease risk. So this can be a whole host of different diseases. I want to make sure you hear something though. All disease risk is not caused by our genetic information. The environment plays a really large role as well. And as we look at different diseases, for some diseases, it's primarily genetic, and for others, it's primarily environmental. So there's a blend in between. Certainly, the genetic information plays an important role in helping us understand disease. So, there we go. Next generation sequencing. This is how we actually read all 3 billion letters. Historically, 1990s, we embarked on the Human Genome Project. Took us 13 years and $2 billion to sequence one genome. Today, machines like this take not quite a week. In 2000, it cost $100 million. Today, it's about $10,000. This is Moore's Law. This is the cost to sequence a human genome. Yeah, major shift. So we're now at the point where we can begin to ask these kinds of serious questions, because it's estimated by the end of the year we're going to sequence 30,000 human genomes. Most of these are going to be in the field of research, but let's talk about why would I need to know this? What can I gain from this information? So we're going to start with a little bit of history. You can trace human history looking at genetic signatures. Now, you certainly could do this by, when we're talking about great-grandma and great-grandpa, but I'm actually interested more in deeper human history, like putting together the family lineage of King Tut by sequencing bits of his genetic information and genetic information from other mummies from around the Valley of the King. Turns out King Tut's mom and dad were brother and sister. But not so uncommon. Not so uncommon if you are trying to consolidate power within the royal family. We also learned that King Tut had malaria when he died because we found the malaria parasite, the genome of malaria, in his bones. Really interesting history pieces. Let's go even further. We can track the migration of human population. 30,000 years ago, a band of individuals moved out of Africa and began to expand across the world. And as each subgroup took off on their own journey, they carried a subset of genetic variation with them. So we have these almost unique genetic fingerprints that let us look at any one of us and get a sense of where our deep historical migration came from. This is not my great-grandmother came over on the Mayflower. This is 28,000 years ago, my ancestors migrated across the Bering Land Strait, that kind of stuff. Let's go back even further. Anybody know who this is? Neanderthal. So this is a National Geographic reconstruction of Neanderthal. They were able to sequence the Neanderthal genome. It was all in tiny fragments. They had to assemble and put it all together. They learned that Neanderthal likely had green eyes. So the picture for this had already gone to press to be on the cover of National Geographic, and they had to computer alter it to make the eyes not blue but green. They also learned Neanderthal likely had red hair, and we can do comparisons back and forth. All right. The world that I live in spends a lot of time looking at genetic variation to understand the cause of disease, to try to find those changes in our DNA that increase or decrease disease risk. And one area where you see a lot of this is in the field of cancer. We can now take a patient's tumor and we can sequence the genome of that tumor and sequence the genome of the normal cells from the same patient. Cancer is primarily genetic. It's driven by mutations in our DNA, but those mutations are acquired through environmental influences or just through random chances our cells divide. So I can compare my normal and my tumor genome and find the mutations that have driven this cancer in this patient, which also means I can begin to tell how likely it is to reoccur or metastasize. And I can begin to move even further to treatment and identify the right kind of medications that actually will be used to treat this cancer based on this patient's genetic background. This is incredibly powerful and exciting. Now, let's back out a second. Back to our genome, and let me tell you some stories about this. Nick Volker. Nick Volker was this really energetic toddler, lived in Wisconsin, began to develop abscesses throughout his abdomen. Turns out that every time Nick ate solid food, holes opened in his intestines and the contents spilled out into his abdomen, and he developed massive infections. This little boy had 100 surgeries by the time he was 4 and continued to get sicker. The only treatment, the only thing that seemed to help was massive doses of immune suppressants, which led the physicians to think this is probably an immune disorder. Ideally, the way we want to treat this would be with a bone marrow transplant. But we're not just going to offer a bone marrow transplant to a 6-year-old until we have a pretty good sense that that's what the cause is. So, at that point, the physicians in 2009 made the really unusual decision to sequence Nick's genome. And in this case, let's go back to the cookbook analogy. They don't sequence the whole cookbook. In this case, they just sequenced the recipes. It was easier, it fit the technology at the time, it was a lot cheaper. Here's the important piece. In sequencing his recipes, they found 16,000 changes in Nick's DNA that weren't in the reference. So imagine that you're the bioinformatician that has to look at that and decide which of these changes actually cause disease and which ones are just part of normal variation, and which ones are that swan's down versus white lily flower substitution. After a lot of bioinformatics work, they found a single change in an immune system gene. And they had found other patients with mutations in this immune system gene that didn't have the same symptoms Nick had. Nobody had ever been seen with Nick-specific symptoms. But these other individuals were treated with a bone marrow transplant. So Nick, at the age of 6, underwent a cord blood cell transplant. That's a lot like a bone marrow transplant. Here's Nick today. He just turned 7. He loves to ride his four-wheeler around his backyard, and his favorite meal is a milkshake and a steak with lots of steak sauce. When he turned the corner after his bone marrow transplant, his mother wrote on her blog, "The heavens have opened up and steak sauce is raining down." Nick's life was saved because the researchers and the physicians sequenced his genome and found the mutation. We're going to see more and more of this, but here's the challenge that comes with this. Well, actually, here's not the challenge. Here's the encouraging part. You can see the cost dropped from $75,000 to $7,500 from the time they sequenced in 2009 until today. Here's the problem. How do you interpret all this variation? And how do you quickly identify the changes that are clinically relevant and the changes that aren't? You know, I told you with Nick, we sequenced Nick's recipes. If we sequenced the entire genome and all that contextual information, we wouldn't find 16,000 variants. We would find about 2.5 million. So this is a huge challenge for anybody in the field. This is where the next big hurdle is that has to be overcome. We figured out the technological pieces. We can do it in a short period of time for relatively cheap, but the assessment is the next big issue. So this really isn't even an appropriate image. We are not searching for a needle in a haystack. We are searching for a specific needle in a stack of needles. That's the trick. Now, there are a lot of people that firmly believe that within the next generation, we're going to see genome sequencing as a standard part of medical care. There are insurance issues that have to be dealt with. There are other questions that we'll get to in just a second. But the technology exists that instead of today doing a heel stick on a baby and doing what we've often called the PKU test, which is really newborn blood screening, that we'll take that sample of blood and we will sequence the genome of that infant. Now, will we analyze all of it at birth? Probably not. Probably we'll just look for a specific subset of genes that we know influence childhood diseases. And then maybe a little bit later on, we'll look at another set. That's the concept of sequence once, analyze multiple times. Let me take it another step. We now know that fetal DNA floats free in the mother's bloodstream when the mom is pregnant. It's cleared as soon as she gives birth, but while she's pregnant, the fetus's DNA is in her blood. So we could non-invasively take a sample of mom's blood, sort out the fetal DNA, and sequence the fetal genome, instead of having to go through amniocentesis or chorionic villus sampling, which is invasive and has a risk of miscarriage. So potentially, even before birth, you would have some of this genetic information. Now, as you might imagine, there are some people that are really excited about this. And there are some people that are not so excited and are filled with questions. And clearly, in the 5 minutes I have left, we are not going to cover those questions. But I want you to think about who gets the information, how much information is given when, who pays for the information. What does it mean to now suddenly have this whole host of genetic information when most of us don't even understand how to interpret our own cholesterol level blood tests? Do our physicians understand how to teach this, how to explain this? No. They don't. I taught medical school before I came to HudsonAlpha. No, it's not happening. So there's a huge educational piece, plus all the ethical challenges that we're going to have to deal with. You can explore your own information. There are lots of companies out here. I am not going to endorse any specific one of them where you spit in a tube, send it off, and they give you back about 1 million bits of genetic information. This is not sequencing. This is more like spot checking, specific places where we know there's variation. Here's the key question you want to ask. Is this medically actionable? Are you going to get some information back? I tell people this is a lot like me giving you a 5,000-piece puzzle, but I've only given you 500 pieces of the puzzle, and I haven't given you the box top, and you now have to tell me what the puzzle looks like. We've got a lot of genetic information that we know, but there are lots and lots of gaps that we don't. So this is tricky. So it really is, take it with a grain of salt. Now, in the interest of being secure and aware, there we go. And again, All right, in the interest of being secure and aware, let me give you a little peek into my genome. Let me tell you a little bit about me other than what you can see. So, here are 3 either genes or these RS numbers are specific locations in the genome. They may not be in a gene, they may be near the gene. And here are my A's, T's, C's, and G's at that location. For most of them, you'll notice I've got 2 copies, one that I inherited from mom and one that I inherited from dad. Now, this genetic information at this location means I have wet earwax, not the dry, flaky kind. This variation, all kinds of things you want to learn about me. This variation means that I am lactose tolerant. This variation right here, this is one of my favorites. This means that if I eat asparagus today, when I go to the bathroom tomorrow, I will smell that I have had asparagus the day before. Please do not tweet that information. All right, let's look at this. These are 2 genes. These are 2 regions that vary. This one actually happens to be in a gene called the androgen receptor on the X chromosome. I only have 1 X. I got it from my mom. My dad passed me a Y chromosome. That's why I'm a male. If I had 2 X's, I'd be female. So this is the androgen receptor, so I've only got this that I inherited from my mom. This combination, according to the company that I worked with, says that I have a very, very, very low chance of developing male pattern baldness. But my barber will disagree with that. So this brings up the point, we don't have all the information. We don't know all the genetic causes, and there are environmental pieces that we aren't able to identify either. So I want to make sure that you don't walk out thinking, ooh, if I get my genome, I know everything about me. No, no, no. Type 2 diabetes risk. Now, let me show you. Men of European ethnicity, as am I, on average, about 26 out of 100 will develop type 2 diabetes. These are 11 regions of the genome that science has shown us play a role in the development of type 2 diabetes. And based on my specific inherited variants, I have a almost 1.5-fold increase. So, men with my specific genotype, 37.4 out of 100 develop type 2 diabetes. This is important information. I want to know this. I'm going to think twice before I take a second Krispy Kreme glazed donut. But there are a lot of other issues I've got to be aware of. My environment, like I just said, this isn't all. All right, let's finish up. Drug sensitivity. This is an area where I think we're going to see big value. This top one, this variant, tells me that I am very sensitive to warfarin, also called Coumadin. It's a blood thinner. So if I take Coumadin, if my doctor prescribes it for me, we need to start me on a much smaller dose to prevent my risk of bleeding to death. Important to know. This one right here, this variant, says that if I'm ever in Europe and I develop a staph infection and they want to prescribe fluoxacillin, I need to run screaming, because this specific genetic variant gives me an 80-fold increase of toxicity in my body if I take fluoxacillin. So this is important information. So that wraps us up. We've covered a whole host of reasons why you might want to learn about the genome, how it helps us with history, our health, and a little humor thrown in the way. You're going to see this in your life. This is going to impact you and your children in myriads of ways. Educate yourself. Learn about what this means. Don't fall victim to the idea that your genes are all that make who you are, because we are so much more than the DNA that we inherit from mom and dad. Thanks a lot, guys. I really appreciate spending time with you.